how regularly the classification is updated for them, and why it is necessary?
It is necessary. The point is that we are constantly learning more about these organisms. We are constantly learning more about different features of these organisms and about evolutionary links and evolutionary, their role, how evolutionary position. So based upon that, they keep on updating this. For instance, when I told you that haplopoda Leptodora kinti was considered as a cladoceran as and just recently in 1990s, in 2000, it was considered as a cladoceran. But now they say no, it is not a cladoceran because it has got an intermediate developmental stage like a nucleus of a copepod. So they change the position and they do a bit different analysis. It is not done in a certain periodic interval. It is done constantly. That's all.
Thank you, madam.
Thank you.
One more question. You said that is the sexes are separate. Is
Mhm.
there any intersex or the sexes are separate?
Yes,
Yes.
intersexes, the sexes are separate.
Yeah.
There are males and females. The
So
intersexes cannot reproduce.
okay. Sexes are separate. So there is any external morphology or sexual dimorphism?
Yes,
Is there?
yes. The sexes are separate. I'll show you if you allow me to share my slide once more. I'll just show you. If you can see this slide, can you see this slide?
Yeah.
And you can see this and the antennules here.
Yes.
They are very long and smooth, and this is a female. And in a male, you will have a kind of a joint, like an elbow joint here. This is used for grasping the female. So this is a very clear sexual dimorphism that you can see in calanoids and in cyclopoids, even in herpacticoids. And I'll show you for males, for daphnids. For instance, over here, you can see these structures. And in Daphnia, the antennule also over here is very long and it is used to capture the female. It is much smaller in size. You can see the size difference. This is an adult male and an adult female Daphnia. So there is a lot of, yes, it is very distinct. Now, if you see over here, the intersexes that you were asking, you can see clearly over here what happens. You see here that this is a normal, here you have a male. These antenna, you see this part is smooth and this part is like a male.
Okay.
So this is neither a male nor a female. Okay.
Thank you, ma'am.
Thank you very much. Any other questions or any questions for Dr. Sarma?
Any other questions? If there is no questions, I would like
excuse
to...
me. Excuse me.
Sure, Akash. Yeah, Akash, you will ask the question.
In a particular season, is there a particular season for copepods in which they thrive abundantly?
It depends upon the species. It depends upon the system where you are working, and it depends upon each species. But more than, I mean, unless the temperatures are extreme, more than temperature, it would be the seasonal development and reproduction of fish. So here in India, you have fish that reproduce in specific seasons. And in the season in which you have high density of fish, it is possible that you will find low densities of Cladocera, Copepoda, Rotifera, because the fish larvae are consuming them.
Okay, thank you, ma'am.
Thank you.
Any more questions?
Any question for Dr. Sarma?
If there is no question, should we continue further?
Yes, please.
I suppose there is no question now.
Mhm.
So, on behalf of all the participants, I am glad to be standing here to express my vote of thanks. A special thanks to our chief guest, Dr. S. Sarma and Dr. Nandini Sarma for accepting our invitation and for being there with us today. And your thoughts have truly inspired us. And thank you, ma'am, so much for telling us about your university and for all the guidance and invitation to our junior researchers. Definitely, they will take this opportunity. And thank you so much, ma'am. So we proceed further. In the next part of the webinar, we have introduction to the guest speaker, Dr. S. Sarma. For this, I invite Dr. Pallavi Sinha,
Thank you
Assistant
very much.
Professor Zoology. Pallavi Sinha. Pallavi Sinha.
Sir, shall we continue or you need a break?
Just 5 minutes, please, because I rearrange few things, and then in 5 minutes we'll start.
Yes,
Okay.
sir. Okay.
Okay.
Thank you.
Thank you.
मेरा तो गायब हो गया लगता है। तुम्हारा ही काम है, तुम बंद
Sir, please unmute if you are ready. And then you can start. Pallavi, unmute yourself.
A very good morning to you all. It gives me immense pleasure to introduce Dr. S.S. Sharma. Although Maheshwari Madam already introduced him during keynote lecture, but here I am giving a short introduction, especially to those participants who has joined the session late. Dr. Sharma is an eminent aquatic ecologist who obtained his PhD in 1988. Dr. Sharma is known for his research contribution in the field of freshwater zooplankton. At present, he is serving as a professor in the National Autonomous University of
Pallavi, you are unmute for you.
He has published a number of research articles, more than 300 numbers in many journals, as well as book chapters. He has edited and co-edited 14 special volumes of prestigious publishers, including Springer, Elsewhere, Francis and Taylor, and he is also a reviewer of many journals, such as Springer, Wiley, and Elsewhere. Presently, he is editor of Rutherford News, which is a virtual newspaper newsletter. He was on editorial board of dozen of standard journals, including Aquatic Biology, Frontiers in
I think there is a network problem in college.
He's a reviewer of research projects national funding agencies. He has guided about 75 students for the award of their thesis at the different levels of education. With this brief introduction, I will like to invite Dr. Sharma, who will deliver his talk on the topic ecotoxicology with emphasis on xenobiotics and cyanotoxin. So please start your lecture.
Thank you. Thank you, Dr. Pallavi, for your kind words. I will share my screen. Can you see the screen?
Yes, sir.
Thank you very much for your kind words. And then this is something, the first one of the two aspects, applied aspects of the zooplankton is the ecotox. Second part is the first part is ecotoxicology. Second is aquaculture. So in this part, I will deal with only basic aspects. The next topic will include how the zooplankton collected from the field can be directly used for as indicators of the water quality. And there are many different types of indices that have been developed, and we will use them in the next talk. Here, I'm going to talk about basic aspects of ecotoxicological application, not only of rotifera, but also zooplankton. So majority of the characteristics I'm mentioning here are also useful for other groups of invertebrates, mainly the crustaceans too. So here I would like to mention something about xenobiotics and the cyanobacteria. These two are more problematic now than in the past. Xenobiotics mostly are those that are not present in nature, are created by, developed by humans. And the topic has got three aspects: one, experimental, and both experimental, laboratory experimental, and field studies. And what are the parameters of zooplankton that we can use as a sensitive indicators of stress? This is second. Third is what are what we have been doing in Mexico. Mexican researchers, what they have been working and how that can propel the ecotoxicological application of zooplankton, this aspect. So some basic aspects, some of it you already have listened to Dr. Nandini too. And these are the basic groups, ciliates, protozoans, majority of them protozoans, and then we also have flagellates and so on. Then you have rotifers, cladocerans, and copepods. These three groups are more important, considering although we have very high diversity of rotifers, of ciliates. For example, we have nearly 1,000 species of non-parasitic, non-pathogenic ciliates. But the use of them is more complicated. Some of them are very resistant too. So I will concentrate on the zooplankton. So these are applicable for rotifers and cladocerans. First of all, they have wide distribution. Wide distribution does not mean cosmopolitan distribution. So many commit this mistake, especially the youngsters commit this mistake, considering that wide distribution means cosmopolitan. No, if a species is found in India, if a species is found in China, do not constitute this one as cosmopolitan. For a definition for cosmopolitan in zooplankton, it must meet with one characteristic, that is the distribution must be, the species must be found in at least 3 continents. So Europe and North America does not constitute a cosmopolitan species, nor Asia and Australia also does not constitute as cosmopolitan. This is very important to distinguish. Wide distribution doesn't mean wide distribution means here I would say that they are found in different places, but I don't comment on continents here. They have there are about 600 species of Cladocera, about 2,000 species of Rotifera, and both these groups reproduce by parthenogenesis. Therefore genetic uniformity is possible. So your experiments do not suffer from intra-replicate errors. Errors will be minimum, or the deviations will be minimum. Both of them have short generation time, as Dr. Nandini mentioned, that generation time of Moina can be something a week. But generation time of rotifers is more or less the same. They have high population growth rate, that is population growth rate is 1 per growth rate is 1 is considered to be very high. And they're small, so that many individuals can be used in the small container. For example, take 100 ml beaker, and you can introduce nearly 500 individuals of rotifers or 500 individuals of Cladocera. So it means when the sample size, when the number of individuals in replicate is high, the sample size becomes high, then it is statistically viable, and the statistical resolution will be high, and intra-variability can be, intra-replicate variability can be minimum, this one. And easy to culture and very cheap to grow, that Nandini can mention about this one in the next topic, how they can be grown on various substances, including wastes. They are also sensitive indicators of change. Small change in pH, salinity, temperature, or even food quality, they immediately respond in few days time. You do not have to wait for many days, for many weeks or months. And they also, their variable, variables we use, their biological variables are quantifiable. So these some of the characteristics that we use them. In case of the copepods, also we have great advantage. They have 6 nuclear developmental stages, and each of the stage can be very sensitive. However, we normally do not use adults of any of these 3 groups, neither rotifers nor crustaceans, because adults are not sensitive enough. So if we consider any water quality criterion based on the adult, say certain concentration 1 milligram per liter of a substance kills adults, much less than that concentration kills neonates. So we need to use sensitive stages in the life history of these organisms. So what is the diversity of these 3 groups? in the world level and in Mexico. We have 2,000, more than 2,000 species of rotifers in the world, and clouds are about 600. Copepods, these are freshwater. Marine copepods are different; they are much more diverse. So we have about 2,500 species of freshwater copepods, and ciliates also 1,500. In Mexico, we have about 400 species of rotifera have been reported. Cladocera now we put around 150, and copepods are around 120 or so, and ciliates nearly 1,000. But you see the last column, 35, 24, and question mark, these indicate that these are the species that have been cultured or that were used once, at least as bioassay organisms. The number is very low. Cladocera, out of 600, only 20 are used. Rotiferous, out of 400, only 35. Sporadic, it means there is no continuity of their utilization. And ciliates, of course, we do not have much information. Worse is a copepod, difficulty to culture them, males and are testing the nuclear, enough number of material nuclear stages. All these are complications, yet it's worth to test them because protection of all these groups is important for functioning of the ecosystem. Imagine that there are no rotifer, there are no cladoceros, there are no copepods in an ecosystem. The phytoplankton remains unutilized. So the energy in the phytoplankton will not reach the fish larva or the next predator stages. Therefore, it is their functioning is important. It's like what we normally describe on in the ecosystem functioning. If it is a ladder, imagine if some steps in the ladder are not anymore there. So the ladder is useless. In the same way, for the proper ecosystem functioning, all these groups are important. But for the species diversity in a given water body, how do we determine? See, this is the species accumulation curve. You need to make several efforts. 1 time, 10 times, 100 times, and filter 1 liter, 10 liters, 60 liters, and go in January, go in February, and so on. Finally, keep on adding species that you have not recorded before. A stage will come where you will not get any more species. That is the saturation point. But this has not been done for many water bodies, including in Mexico, but we know approximately 50 is a more or less the limit that you can expect in a given water body. In other words, if you find, for example, 5 rotifers in a given water body in one attempt, we do not conclude that the water body has only 5 species. So you need to make more sampling, possibly in different periods of time too. So that way we have the copepods nearly 8. This is estimated value based on many different studies, and cladocerans 15 and rotifera. And as I mentioned, not every stage of rotifer is useful for ecotoxicological studies. Resting eggs directly cannot be used because they are very resistant. They are thick-walled, and then they do not respond. Even if there is a small change, that cannot be detected using the tools available in ecotoxicology. For example, small concentrations of metal is absorbed by the rotifer resting egg. A part is adsorbed, a part is absorbed. So by absorption. So in this case, that you cannot detect easily. Therefore, we need to use stages that are sensitive, and they yield certain variables which you can measure quantitatively. So mainly for rotifers, we use neonates. Males are not used. We use to individuals, female individuals, neonates. Males are not useful because rotifer males live only for 24 hours, sometimes 19 hours. Many of them less than a day. So you try to do an experiment for 24 hours. In the meanwhile, the lifespan of males finishes, and then they die. You do not know whether they die due to your stress or due to their natural mortality, physiological mortality. Males of rotifers do not feed. They do not have alimentary canal. They do not compete with females for food. Their only to make resting egg production in the female. Therefore, males are not used, although they are abundant sometimes than females in a given water body, their lifespan is short. So we use only females. Not every female. Individual females of different ages should not be used because they respond differentially to toxic and stress. Some individuals die early, some individuals die after 2 hours, and so on. So this is not useful. Always you must start with individuals of known age. Therefore, intra-replicate variability can be low. Okay, in this case, you can use, however, neonates as coming out of resting eggs. So you purchase the resting eggs, they are commercially available. So you hatch them within 24 hours. The majority of them hatch, about 80% hatch, and then you start experiments with them. They are sensitive. Or if you are culturing in the laboratory without purchasing them, then amictic eggs hatching within 24 within 12 hours, they normally hatch. Many of them, if you have a large culture, synchronous hatching is possible, and use them for ecotoxicological studies. Okay. Which genus is more utilized? Well, the question is, which genus is available for you? Brachyonus basically is a tropical, pantropical species. In temperate regions, you do not find. However, in the laboratories, yes. So you need about 20 to 25 degrees centigrade. And then we have one water body. So you will see that it is a touristic water body. And it's a shallow, it's less than 2 meters. And then it's propelled manually, moving forward and backward, and is not allowed any mechanical boat device because it can contaminate the water body. And in this water body, we find about 15 species of Brachyonus. Therefore, you can use any one of them. All the 15 species are culturable. Fortunately, most of them feed on the alga. Here is an example of different body sizes. There is no relation between the body size and body and species sensitivity. Large species can be equally sensitive as a small species. Therefore, which species we have to culture is dependent on which chemical you are interested to test for. This is the Cladocera, the life cycle Dr. Nandini has already explained. In this case, third or fourth, usually between second, third, and fourth, the clutches, they produce in clutches. Daphnia magna, for example, is a very large species. Each clutch it can produce from each individual 100 or even more neonates for each clutch. So the second and third supposed to be the animal stabilized. Young mothers produce clutches too, but the number is small, and there is some variability in their survival, in their ability to respond to stress. Therefore, second and third or even fourth clutches are generally ideal for this purpose. In copepods, you have more advantage because there is no direct development. Most copepods develop through nuclear stages, and each nuclear stage is sensitive, but it lasts only 1 day or 2 days. So experiments should be done very fast, and long-term experiments are not possible because they mature. Okay, they reach the other stages. So both field and laboratory experiments can be done in ecotoxicology. Ecotoxicology depends on sound principles of ecology. Toxicology, the difference between toxicology and ecology is ecotoxicology and toxicology. Toxicology related to one individual or just one population. For example, if a person consumes one medicine and then dies one particular substance, and the medical report is restricted only to that individual, what has it has nothing to do with other organisms, for example. Whereas in ecotoxicology, we are concerned if zooplanktons die due to some substance, then the consequences are the next level. Or if zooplankton accumulate certain toxic substances, the consequences will be for the next level of higher level in the aquatic food chain. For example, contaminated zooplankton can be consumed by fish, and fish might accumulate what is called bioaccumulation, and humans feed on that, or birds feed on that. So the consequences can be beyond one individual. That is what we call ecotoxicology. However, ecotoxicology relies on the principles of ecology. It cannot exist alone. Therefore, all the standard ecological theory, ecological concepts will be utilized in ecotoxicology. They cannot develop something new, except one or two cases where they are normally not ecologically relevant. Most of the toxicological works are ecologically relevant. One or two cases I mentioned because you sometimes test the effect of microcystins in the rat. Rat population, rat terrestrial, it does not live in the water, whereas microcystis is aquatic. So there is no relation as far as ecological relevance yet. We need to study because humans or animals drink microcystin contaminated water, it will have an effect on their kidneys. Therefore, for predictive purposes, sometimes if toxicological works are carried out which are not ecologically relevant. But toxicologically, yes. Okay. Then, biotics, these are chemicals foreign to organisms, mostly the animal species. And these are pharmaceuticals that are not present in nature, body are modified in nature, are modified from the natural products in order to suit the human body, other organisms, and the pesticides, the cosmetics are exclusively developed for humans, and then the industrial chemicals, and more important now is the microplastic pollution is becoming more and more evident, and it's a hot topic. Any article on that, any manuscript on that is likely to be accepted because we have much less data. Until a few years ago, until 2 decades ago, we consider plastics are neutral and they do not do damage. But now we know that is not the case. It can do damage both directly and indirectly. We will see some data on that. And then standard journals now publish a lot of information, quantitative data on the impact of microplastics in aquatic ecosystems. Then two types of evaluations can be done. You are in a hurry, you get a lot of information, a lot of articles you want immediately, then make acute toxicity tests. Acute toxicity tests are also done by the governmental agencies, and chronic toxicity tests take a longer time. Acute toxicity tests give information within 24 to 48 hours. How they are done? There are 3 different methods to carry out the experiments. One is a static method, that is you collect a certain concentration, add a certain concentration of a substance into the medium, and then expose the test animals, and then after 24 hours or 48 hours, you count how many of them are alive, how many of them dead. From there, using a statistical probit method, you will come to know 50% of the mortality that I will show you within. But before that, I would like to add the difference between concentration and dosage. Dosage and concentration are not the same. For example, dosage means the quantity exactly that enters the body of an organism. Concentration is something that you expose the animal to a known density, a known level of a substance, but you do not know how much it enters the body. When you swallow a capsule, you know how much exactly it enters your body. But when you expose an animal to a medium containing toxin, you do not know how much it entered. This is one of the main reasons why diabetic medicines, inhalers were not successful. There is some attempt to develop, for example, anti-diabetic medicines as inhaler, so that you don't have to take capsule 3 times or 4 times a day. You simply inhale. But the substance is cotton is soaked in the substance, and then the patient is allowed to inhale. But the problem the doctors face is that how much the patient inhaled. A weak person probably does not inhale much as a healthy person, healthy but diabetic problem. So therefore, the quantity that enters human body is not quantifiable, is not known to the medical doctor, so he cannot give a proper prescription for you. Therefore, this is still developing some time. In the same way, you expose the animal to outside, you do not know how much concentration of the substance entered, how much volume, how much the quantity of the substance entered the animal body, you do not know. So that is why we call it the concentration. We cannot use doses for the dosage for the animal because you cannot inject except in the mammals, yes, but not in the smaller organisms, especially the rotifers or even cladosaurs. You cannot inject. This is a difficult step. Therefore, we still expose the animals in nature to the concentration of a known substance at known density. Okay, if the static method is simple, as I mentioned, you expose the animal to a concentration, and then after 24 hours, take them out and then count how many of them dead or how many of them alive. The second method is static. One of the methods in the simple static method is that oxygen concentration changes, because first hour there may be full oxygen, second hour there may be depletion, or the animals that live there start excreting some ammonia and other products, so they change the water quality. So every 24 hours or 12 hours, you can change that. Semi-static or a static renewal system is the most commonly used. Now, a third variety, much better than the third method, much better than remaining two, is the flow through. A constant concentration present in the medium is circulated, and the animals are constantly exposed to this. So there is no loss of any substance. There is no inclusion of any excretory products. But one problem is it is very expensive. And for doing a small 24-hour study, this can be costly too. Now, there are three terms that we commonly encounter in ecotoxicology: LC50, EC50, and LT50. LC50 is a lethal concentration 50, where 50% of the test population is expected to die. EC50, where animals are not dead but dying. Death is an event, dying is a process. So the process leads to the event. However, the individuals that are dying, that are not responding to needle, when you try to touch it, the antenna does not move. It is definitely going to die. Even if you return the animal to the clean water, it is not going to live. So then that is considered to be, in many cases, whether it is dying or dead is difficult to detect, especially the smaller animals. The same thing also for humans, probably. Clinical death is different from other kinds of death which we consider. So some organs may be still functioning, where the brain might be dead. So in this case, when an animal is not responding to a touch by a needle or a brush or anything, and this considered to be dead. Therefore, this EC and LC in some cases are identical. Now, LT is a lethal time. When concentrations are extremely low, the animal doesn't die in 24 hours or 48 hours. Maybe it requires more time. So the concentration is the same, but in different times they will die. This is based on the observation. Every 24 hours you observe, don't change the medium, don't change anything, leave it there. But how many individuals, they're starting how many, and at each day, after each 24, after each day, how many of them are dead. And based on that, we can derive LT52. These are normally done for industrial product. When industry produces certain toxic substances, very low levels they introduce into the medium, into the natural water body, that does not kill any of our test species. So we consider that is safe. It may not be safe. It may be safe for 24 hours, but not for 72 hours, for example. So in this case, we can't ask the industry to enhance the toxic substance because our animals are not dead. This is just not possible. So what we do is we collect the natural water and expose the animal, our test animal, and keep on checking how many individuals are alive at a particular time. So concentration is constant, time is variable. In LC50 and EC50, time is constant, but concentration is variable. Okay. The sublethal concentrations are more important, many times more important than the lethal concentration test. Here is an example. I consider species A and B. Both of them have identical LC50. That is, both of them die when 50% of the populations of both species will die when you expose them to a concentration of 1.5 milligrams per liter of an unknown substance, for example, of a specific substance. Now, but if you want to see 20% of the mortality, you extend the graph, and then you will find species A is dying at 0.5 grams per liter 20% of the population will die, whereas a species, that is species B, and species A, 1.1 milligrams per liter species, 50% of the population, 20% of the population of species A die. In other words, species B is more sensitive than species A at sublethal concentrations. It is very simple analogy. For example, you take 10 persons of the identical body weight, but they run differently. All of them don't run with the same velocity, but weight is the same. Here is the same. The mortality as 50% is identical, but at sublethal levels, their performance is completely different. Therefore, you consider the species A and B will be protected by a concentration of 1.5 milligrams per liter of toxic substance. At sublethal level, it can be completely different, and one species might die much earlier than the other. 1.1 milligrams per liter, if you expose this concentration to species B, it dies much earlier, more than 20% will die. Therefore, in many ecological works, LC50 alone is not sufficient, is necessary but not sufficient. You need to proceed further. Now, some of you have asked, Nandini, what is the ecological role of zooplankton? Here is that most of these tests that are done ecologically relevant are also ecotoxicologically relevant. For example, both field and laboratory studies, you can study somatic growth with and without toxin. You can study body size, egg size relation with or without toxin, rate of respiration, swimming speed. Swimming speed decreases with increase in toxicant concentration. Functional response, how much food they eat. The food consumption decreases with increase in the toxicity. Then bioenergetics, how much energy is actually collected by a rotifer or by a cladocyrene or by a copepod, and how much is transferred to the next level. Okay. Then starvation studies. Rotifers normally do not live more than 2 days without food. But if you have toxic substance and no food, they will die within 24 hours. So it is a double stress. One is a starvation, another. So these aspects are short-term experiments, but can be done, and they are relevant in most cases to quantify the effect of toxic substances. Now, similarly, mesh size. What mesh size do you have to use? In metal contaminated water bodies, generally the body size is smaller. So of the zooplankton, then you can have a mesocosms, microcosms. Then you also have a microbial loop where together with detritus, they have a functional role. There's something like a facilitation means the excrete of one animal is used as a food for animal. If you culture the one species, for example, ostracods, the excreta is good for the rotifers. Some of the rotifers cannot eat directly the food that ostracods eat. It can be macrophyte leaves, for example, but they excrete out, and these excreted products are partly digested and useful for the rotifers to grow. So any substance that kills this process will kill the entire, will kill the species in the chain. In the food chain can also kill the process. So then we have a competition, two species competing for the same food. When a toxin is added, one species sensitive, it is wiped out of the competition, another species dominates. This is what normally happens in the contaminated water bodies. One or two species dominate, whereas all other species, they disappear. Now, we also have behavioral studies. We have life table demography studies in the laboratory. You have numerical response, and you also have predator-prey relationship. Finally, you have allelopathic relationship, where the macrophages usually produce some kind of substances. These are not necessarily toxic, but these substances will not permit other phytoplankton, other species, other primary producers like alga, they do not grow, or cyanobacteria even. So allelopathy reactions are the best example of allelopathy is the penicillium. The penicillium, the fungus, where it lives, it produces a kind of substance, penicillium. Now we know penicillin, the same substance we have identified, and around the bacteria get killed. How are they killed? They are killed by the chemical substance produced by the fungus. Okay. These are some of the processes that in the toxical process that have been going on in ecosystem. Many of them will be disrupted, not just one. with the presence of just one toxic substance sufficient to cause these lot of sometimes irreversible changes. Now, what we can measure in the chronic test, it test morphological responses. For example, it's only morphology, not morphometry. You can also check the morphometry. Morphology means a malformation of lorica, malformation of some spines. Some are Nandini already showed some intersexes in case of copepods. These are all due to some chemical substances. Pesticides, herbicides cause this kind of morphological changes, and you can quantify them. When you quantify them, it becomes a morphometry, and morphometry. You also have now morphogeometric methods that not just only morphometry, but geographic spatial position can also be measured. Then you also have somatic growth. Most rotifers reach maturity within 24 hours. If you add a toxic substance, instead of reaching 24 hours, they take more time. Therefore, they all these variables were done for different species, but not the same species. Here is a body size variation we measured. I exposed the same population, started with the same individuals of the same age, and in neonates we measured they are the same size, but after 24 hours, this was done after 48 hours. And when you expose them, see the heavy metal contaminated 1B became smaller than the control which we have A, they're slightly larger. So these differences are statistically significant. Then this is the example that at different temperatures, they have different growth, body size. And when you expose them, when they are growing, expose them to this toxic cyanobacteria. This is an example Dr. Nandini had done many years ago. And the Cerodaphna cornuta at 20 degrees and 30 degrees, two different temperatures they exposed, and controls the body size much longer than those exposed to microcystis. Microcystis is a cyanobacterium. When the cells were exposed, when given as a food, Cerodaphna consumes the toxic cells too, but its growth rate is the body size is much smaller. We do not consider the body size small because there's a different species. It is the same species, the same population, but exposed to different toxic substances. The differences are statistically significant, as you can see. And we also can find the interrelationships between body size and body weight, body length and body weight, and so on, different parameters. This regression equation changes, the shape changes depending on the stress. And behavior, some of the species, for example, Brachynes rubens, it has a tendency to attach itself to cladocerans. So it doesn't have to swim. And the cladoceran swims, automatically this animal also swims without any effort and collects the food as cladocerans swim. This is their tendency. But if you add substance to the medium, immediately they detach themselves. They start swimming normally because they don't like the toxic substance, but they are within the medium. Then the swimming rates, as I mentioned, that normally rotifers swim at the rate of 1 millimeter per second. And if you add any toxic substance, the swimming speed falls down to all the way half a millimeter or 250 micrometers per second. And somebody has also asked the question about the ability, the vertical migration. Yes, they do, rotifers and cladocerans, copepods also do. But in the presence of toxic substances, this vertical migration, this response disrupts, and some of them migrate, not same percentage as in control. They have different responses. These responses are mainly due to the toxic substance. And male-female encounters is another part. The male alone is exposed to toxic substance, or female alone is toxic substance. The male-female encounters are disrupted, and therefore resting eggs are not produced. The resting eggs are not produced in a water body, and if this water body dries up in the summer, then you have no more individuals left for the next generation to start when rains. Then age at maturity is another one, as I mentioned, that 24 hours and any toxic substance can delay oxygen consumption rates and changes to molecular level. Also, for example, heat shock protein, you can easily measure with reference to the rotifers. What about the physiological responses? Yeah, one of them is hatching time. Most most parthenogenetic eggs of rotifers hatch in less than 24 hours time, but a medium containing heavy metals or pesticides, sometimes the hatching rate is also low and hatching time also will be very high. And then you can do this one based on the survivorship curves, which are based on the parthenogenetic eggs or even those obtained from resting eggs. Here is an example how hatching time decreases with increasing temperature, and hatching success also increases at optimal temperature of 25, but slightly higher at 35 degrees centigrade. So optimal for tropical species, low temperature is not good. In this case, when you expose the eggs to 15 degrees centigrade, the hatching success falls to 50%. But the same thing, nearly 90% or even higher, parthenogenetic eggs in general hatch 100% compared to resting eggs, which require a special stimulus. Therefore, their hatching rates are generally low. Now, this is a survival. You take a population and then expose them to a known toxic substance or even cyanobacterial extract, food extract, or even cyanobacteria directly. The survivorship curves, as shown here, there are normally three types, 1 and 2. Type 1 is very little mortality until they reach nearly half of their lifespan. Then they start dying due to physiological aging. Type 3, which you observe, the several individuals born, they are very sensitive, so they die immediately, and only few individuals resist, and then they continue to live and finally die due to physiological reasons. Whereas mostly the birds don't die because of aging. They die because ecological factors reigns. They do not have proper protection themselves. So this is the type 2. Type 2 is not common in laboratory conditions. Type 1 is the most common non-stressful conditions. And type 3 is very common in stressful conditions. Here are different examples. When chlorella and Senidesmus, two edible food items are offered, the mortality pattern, this survival pattern is different. And age-specific expectancy is another variable that we can easily measure in the laboratory, starting with a cohort population of some individuals of known age, of known clutch, and then start following them. And usually this is the age, as the age advances, the expectancy decreases. So newly hatched individual can live for 12 days, but 7 days old can live only for 3 days because this is normal. This behavior changes, this curve pattern changes depending on the test conditions. Most rotifers and cladosters do not have some copepods also post cessation survival. That means after stopping of the production, normally they die. Some of them die when the eggs are still developing because they exhaust their energy and nothing is left. However, some species continue to live beyond cessation of reproduction. In this case, as when rough estimation provided by King says that about 33% of the zooplankton still live even after reproduction. Reproduction stops, for example, after 3 weeks, then they live for 1 week more. No more reproduction, but still feed on the food item available. So one of the researchers, King argues that if the animal reaches 50% of his lifespan, that is a medium lifespan, if it reaches, then its contribution to the population is negligible or non-significant. Therefore, if it lives or dies, no problem. In other words, if a rotifer or a cladosaur or a copepod species lives, for example, 30 days, 15 days is considering its median lifespan, and whatever maximum production high output will be in the first 15 days. Remaining 15 days, whether it lives or dies, it doesn't contribute to the population significantly. That is his argument. So at the same time, there is also, based on this, he developed a hypothesis which is worth testing in the laboratory cultures, that mean lifespan and generation time. Generation time, half the mean lifespan. This is the hypothesis in heteroparous organisms, that is a species that reproduce continuously, not one-time reproduction, that is semiparous. Heteroparous organisms means the species that reproduce in various batches. Most animals produce various batches too. So he thinks mean survival time, for example, is 12 days, then generation time is 6 days. If a population lives for 50 years, that's the mean lifespan, and the generation time is 25 years, according to this hypothesis. It is valid for many cases, but it is also indicator of stress. When you consider temperature as a kind of stress, at 35 degrees centigrade, you will see about 22% the deviation from the expected relation of 2. So at 25 degrees, the deviation is nothing. It's almost 0.04. It's not significant. So under optimal conditions, yes, that relation holds true, but suboptimal or under stressful conditions, this relation fails. This is also one of the important variables that we use. Edmondson developed an indication of the water quality or the status of the health of the individual of rotifers or even cladostridens and copepods too in natural water bodies. Then now you see one rotifer with 3 eggs. Right now, the population density is one individual only, is 1. Tomorrow, all the 3 hatch. So the population density for tomorrow is 4. Therefore, whatever you see now, the relation reflects in tomorrow's growth rate. So he developed a growth rate based on the egg ratio, the number of eggs by the number of females. This egg ratio changes with the population density. When the population density is very low, egg ratio is very high because there is abundant food. But as the population density increases, per capita distribution is low. When the population density is high, egg ratio is low.
Yeah, it's a request to all the participants to please unmute your mic. Aruna Shrivastava, please unmute your mic. Sorry, mute your mic.
Yeah, I have time finished or some.
Hello.
Okay. So I think. Okay, I can continue, right?
Yes, sir. Please continue.
Okay, thank you. So egregious also decrease with increase in the population density. This is based on the field observations. So, to test is the cost of reproduction. Reproduction is a costly process, energetically very expensive. All animals have to undergo this cost. So there are 2 types of cost. Today's reproduction will have an adverse effect on tomorrow's survival of the population, or today's excessive reproduction will have an adverse effect on tomorrow's reproduction. This can be easily tested, very well known in many. For example, unmated rats and mated rats, take them. Rat will live longer than a mated female rat. It's also true for the copepods, also true for the rotifers, also for the plants. So if you remove the inflorescence of the plant for over a very long time, every time inflorescence are cut it off before it comes out. So the plant lives longer because its energy is retained in the body. So under most favorable conditions, the animal after reproduction recuperates a part of it. Yet, for each reproduction, a part of the maternal tissue is damaged. Therefore, it will have an adverse effect on the future survival of the mother, or future survival of the offsprings, future production of the offspring. So there are two tests that can be done easily using
Sir, just a minute. I mute all the participants and then you unmute yourself.
okay.
Just a minute. Sir, now unmute yourself.
Okay. Yeah. Okay. Can you hear me now?
Yes.
So this reproduction is detrimental to the survival of the mother. This is the concept. This is what we call cost of reproduction. It can be reflected in two different forms. One in the survival of the mother or production of the future individual also. So these two are successfully tested, and this is still considered as a hypothesis because when the mother is well fed, or conditions of the mother are adequate, nutritionally adequate, even if the mother loses certain quantity of energy in the reproduction, mother can recuperate. Therefore, it is considered to be a hypothesis, not a theory. It is a hypothesis. It can be possible in some cases. It is not possible in some other cases. Stress can cause more problems on this one. So there are two methods. You make simple correlations between today's reproduction versus tomorrow's survival, or today's reproduction versus day after tomorrow. The reproduction. So these two can be easily calculated. Usually, individual zooplankton species under stress, when they reproduce, they die faster. Or zooplankton species, when they reproduce today, tomorrow the reproduction will be lower, day after tomorrow much lower until they recuperate. This kind of negative relationship. And finally, you have standard survival tests, and then survivorship curves, and hatch and longevity curves, and reproductive curves like a gross reproductive rate, net reproductive rate, reproductive cost. These are general generation time and the population growth rate. They can easily be measured using sublethal concentrations where the animal is not dead due to acute toxicity, but slowly the impact will be on the reproductive system or on the survival system. Most variables related to reproduction are more sensitive than variables are related to survivorship. This is the general rule. Okay, then other characteristics in the population growth, you have the rate at which population multiplies can be affected by the toxicants, by the pesticides, or even by the microplastics, because most cases what happens is the animal, mostly the rotifers consume, also cladocerans, they consume microplastic thinking there's a food. So their stomach is full and they don't eat real food. They don't eat edible food, and these microplastics are not food. So they die of starvation. Though stomach is full, they die of starvation. What is present in the stomach is not suitable for giving them energy. So these variables you can always measure. The next factor is prey-predator interaction. So you can expose the predator to a toxin and take it out and offer prey, and its ability to feed will be reduced. Or on the other hand, take the prey, expose the toxicant, and introduce in a container containing a predator, clean container containing clean water, but prey was toxicants, or toxins. Then the predator is unable to feed them because prey normally swims, but in the presence of toxicants, toxic substances, prey cannot swim the same speed. So predator-prey interactions can be easily quantified. Then competition, finally bioaccumulation, biotransfer, and these are some of the other variables. I will take some of this aspect tomorrow too. Then cyanobacteria specifically, cyanobacterial blooms are very common. It's unusual that if you don't see a cyanobacterial bloom in tropical waters, usually in the summer, and it looks green, but the containing looks blue-green, and it contains mostly toxic cyanobacteria microsystem. What are these? What is actually due to this one? of the water bodies. Eutrophication is a process in which it's also a natural process but slow, but is accelerated by human activity, mainly because we release nitrogen and phosphorus through different activities. Here is an example due to humans. For detergents we use daily, we still do not have phosphorus-free detergents in many countries. In Europe, they about 20 years ago, they detergent containing phosphorus. So then you have less phosphorus releasing into the waste waters. And domestic waste waters, approximately we release 50 grams of phosphorus per year and per person. And more damaging is fertilizers and from in the fish farms and agriculture farms. They release at the rate of 20 kilograms because they apply NPK fertilizers, and then in the phosphate, superphosphate also they use. So it's a double phosphate. And then much of it is not taken up by the plants, and excessive is added by the farmers. There very few countries have called drip irrigation or point irrigation, and then normally most farmers in India, they apply the fertilizers, and then after some time they irrigate, and then the fertilizers are washed off into the nearby rivers and lakes. So this is the nutrient source. So what will happen if the excessive due to eutrophication, green algae will be replaced by cyanobacteria, and low oxygen levels will develop because cyanobacteria form the net, some kind of plate-like thing on the foam and very thick layer, and will not allow oxygen interchange of the oxygen in the water surface in the air, and therefore anoxia will develop and fishes die. This is very common, especially the fishes. Then changes in the zooplankton community also change because large zooplankton species will be removed and small zooplankton species can survive, and they are not useful because they cannot kill. They cannot remove the cyanobacteria because they are filamentous. Filamentous cyanobacteria require larger zooplankton like daphnids. Moina is also sufficient to some extent, but the smaller cladocerians like chidorids, they cannot do anything. And much worse with reference to the rotifers, they cannot feed on the large cyanobacterial film or colonies. Then, therefore, algal blooms, green algae will be replaced with the time, and only toxic cyanobacteria alone will survive. And they have a strategy. Over 2.5 billion years since then, they are the best competitors with primary producers because they avoid predators by forming colonies and forming large filaments. As I mentioned that rotifers and smaller cladocerians cannot do anything. Copepods avoid them, but they have no other alternative food, so they try to feed on the toxic substance and then die. So these cyanobacteria, basically three different types of toxins. They produce neurotoxin, hepatotoxin, and cytotoxins. There are also other types of toxins, but they are much less known. Neurotoxins, they can kill the functioning of neuromuscular coordination. Hepatotoxins also have adverse effect in fishes, humans, and then cytotoxin also, they are toxic. So these are three different types of major types of cyanotoxins produced by the cyanobacteria. And this is how they, if you see in the literature for the last 30 years, 40 years, you have information on the crude extracts of the cyanobacteria. You take the cyanobacteria, choose a particular species, and then freeze it and thaw it, and you release by this method you release the cyanotoxins. But in the crude extract, it's not allow cyanotoxins alone. There are many other substances that are there, but they can be directly tested. And we also have tested this accumulation of microcystins, and these microcystins are very resistant. In boiling water, they resist. Nothing will happen to them. So take microcystins and boil for about 20 minutes in the water. Nothing will happen. They just remain. The structure is very resistant. So they also enter your body if you feed on the microcystin. So we have tested the bioaccumulation of microcystin, and you will see the accumulates a huge quantity of in the fish. In tilapia, we tested this one huge quantity of microcystins are present in the liver, and the muscle, fortunately, there much less is accumulated and also variable. So this accumulation of microcystin is also dependent on the season. So one-time collection and evaluation of fish is not sufficient. You need to do repeatedly. Then the predator-prey interactions. Dr. Andini has already explained or will explain. There are different steps before fish larva can catch a prey. These are encounters, attacks, captures, ingestion, and in some cases they reject the prey or even the prey itself can escape. So these are quantitative terms. Here I give an example of an rotifer Brachynes videndatus, which develops the spines against its predator called Asplankna. The Asplankna, there are 4 species. The Asplankna brightpilla is one of them, and this produces a kind of allelochemical. This is this chemical pheromone, we call them, because the animal-animal interaction. And these pheromones attack. The pheromones help the prey to develop spines. Therefore, prey and predator can live together. The substances released by the predator are recognized by the prey, and then they develop spines for the next generation. This is sometimes they call intelligent predator hypothesis. Many people don't believe this one, but it exists. That is, a predator for its own survival produces the prey with spines, with the defense mechanisms. Therefore, it does not exterminate all the prey, and a population can survive still, and of the prey can still survive in nature, and predator can feed on that. This is sometimes they call hypothesis. Now, population growth studies, without studying their demography, you can simply measure the densities of the population. Here are some of the variables that can measure population growth rate, effect population, and the growth rate. As population density increases, growth rate decreases. And peak population density, the peak abundances that we can harvest can also be on the presence of substances present in the medium, the egg ratio, and the sexual reproduction. There are 4 general models of population growth, but zooplankton generally show the logistic population growth, and for some time they show exponential growth as the food densities diminish. Then they finally follow the pattern of logistic growth. Then there is no more growth. Some of the growth patterns that we normally use them, these models use them in the bank for your money. So the carrying capacity individuals keep on growing with their eggs. Eggs do not have, all eggs are potential offspring. So eggs of today do not have any impact in the population, but tomorrow they hatch and become neonates and compete with adults for food. The population shoots up for some time and gradually falls down. A stage will come when minimal individuals are left and they are able to utilize the available food. Again, they keep on increasing. This kind of population cycles are very common in nature. By conclusion, I would like to mention that different parameters of zooplankton are used to detect the adverse effects of toxins. Then there's no single zooplankton species. You might think that we can use Daphnia magna, you might use Brachynes calciflorus, you might use Brachynes patulus. No, there is no single species that can be used as a universal species for consistently sensitive to all toxins. Like there is no universal medicine for all the diseases. This one. Second, there is no single variable. For example, you might consider that the population growth rate as a sensitive variable. It is better than most, but not the most of all, because some species are sensitive only to survival. For example, salinity is one of them, that it has nothing to do with the reproduction, but it kills directly the population itself. The survival itself is threatened. So reproduction is secondary statement. So similarly, both acute and chronic toxicity evaluations are needed. Do not discard one for the other. They are complementary, yet for any conclusions that are drawn based on acute toxicity may not be appropriate for protecting the zooplankton in nature. Therefore, both tests are needed. Thank you very much. And then if you have some questions, I'll be happy to answer them.
I have one question. So
Please.
behavioral aspects changes with species to species is different, or rotifer is the same behavioral pattern?
If you compare between the Cladocera and Rotifera, or Cladocera with the Copepoda, the strategies are different. Because in copepods, you have both males and females.
Okay.
Okay, then what happens is females live longer than males.
Okay.
This is number one. And in copepods, you also have different developmental stages. So it gives additional, most of the studies made on one particular stage is not necessarily protective of other stages, right? So you need to do that. In case of rotifers and cladocerans, they are both of them parthenogenetically. Most of them reproduce parthenogenetically. So depending on the substance that you are using, you need to choose whether you can work on one group of organisms parthenogenetically reproducing, another is sexually reproducing. So our idea in ecotoxicology is protect the environment, protect the extinction of the zooplankton. So if the zooplankton, as you mentioned correctly, also one of your colleagues also mentioned about the role of zooplankton. See, if the zooplankton are missing in an ecosystem, the energy in the phytoplankton cannot reach anywhere higher trophic levels. The fishes will be devoid of food, and phytoplankton keeps on growing and growing, and then nothing can be done out of. You cannot harvest anything. With the time, it collapses the system.
Thank you very much, sir.
Thank you very much. Tomorrow there will be another talk that on the bioindicators. You do not have to culture any animal. Okay. You do not have to culture any organism. Whatever you have cultured from the field, use different methods.
Gunjan, please unmute yourself.
Okay, you can use different. You can use different indices that I will show you, and you can develop your own criteria, water quality criteria. You don't trust the criteria developed in Europe and North America. It's not suitable for India because species diversity is different in India.
झाड़ू वाड़ू रखे धुलता होता है निकल गया होगा हमारे किचन में पानी पकड़ा था डालो ब्लीचिंग काट डिपेंड नहीं
Sir, please unmute yourself.
Yes, I did. Yeah. Thank you very much. And any other question? I think Dr. Renu cannot hear, I think.
Okay, I am allowing, sir. Now unmute yourself, ma'am.
Yeah.
Yeah. Sir, there is one question from my side. You said that resistance X are available in market. Yeah. Okay. So I want to know, it is available in market or commercially available only for the scientific reason, or is there any other purposes?
Any other person can purchase them. Therefore,
what are the purposes for behind the scientific reason?
For example, that you want to, you have a fish farm.
Yeah.
And you have no skill to culture rotifers. So you collect them and hatch them and feed them, like what you do with Artemia. So fish farmer normally is not interested in scientific research. In fact, I worked for some time in a shrimp industry, and they are not interested to publish. So the problem is their problems remain them with themselves. They are never known to the scientific community. So for this purpose, you can purchase another. You have an aquarium in the house, and you want to show children how predator-prey interactions happen. Them and hatch them, and they hatch faithfully. You don't need anything except water. Just put the water, tap water, they hatch. And then you introduce them, and then fishes one by one comes up and then picks up the prey, and then that will be a great fun for the children. So that is nothing to do with any serious scientific work.
Okay. Thank you very much, sir. There is another question. You said that in acute toxicity, the duration is the 24, 48, and 72 hours. Is there any rhythmicity in this?
Yes, there is the point is that if you are doing with roti for 72 hours, they mature.
Okay.
And then they produce more individuals. So you started with the 10, for example, or you started with 100, and then some of them mature and then reproduce another 20. You do not know whether which one died, which one is alive, and therefore the rule is before they reach age at maturity, you must stop the experiment. Otherwise, they multiply. They multiply, they create problems with your initial reading, right? You start with 100, and then after 2 days you 110, and then you don't know whether how many of them are dead. So that is the reason.
Okay, thank you very much, sir.
Is there any more questions from the audience? I think there is no more question. So now I would like to invite Mrs. Gunjan Ojha, Assistant Professor, Department of Zoology, for vote of thanks.
Thank you, ma'am. We extend our sincere thanks to Dr. Sharma for providing his time and sharing valuable guidance regarding copepods and cladocerans, which acts as sensitive stress parameters. How toxicity affect both the animals, both species of animals, how their morphology change, how their reproductive patterns change. He enlightened us very much about zooplanktons and toxicity. I think in future his work should be referred by many researchers so that much of the work can be done in this field. Thank you so much, sir. Thank you so much for enlightening us. Thank you, sir.
Thank you very much for your interest. I see that nearly 70 people have participated, and then actually you have attracted a large, interesting, enthusiastic participants. That I congratulate the organizers for this. Yeah.
Thank you so much, sir.
Thank you, sir.
Let me start with the next lecture of Dr. Nandini.
Okay, she's coming in 1 minute.
Yeah.
Nandini is coming in 1 minute, please. She's adjusting.
Yes, sir, please.
Thank you.
Good afternoon.
Welcome back, ma'am.
Thank you so much. Can you hear me?
Yes, ma'am. It's very clear.
Very long session, very long session. I hope all of you are not very tired.
No, not at all, ma'am. We just are listening. Lecture आप दे रहे हैं.
नहीं, सुनने में भी तो थकान होती है बहुत.
नहीं. It's very interesting. We are enjoying. So many new things we are knowing.
Thank you so much.
So now I invite Dr. Nandini Sharma for her lecture on large-scale phyto and zooplankton culture. Over to Nandini, ma'am. Welcome, ma'am.
Thank you so much. Can you see my screen?
No, ma'am.
No, ma'am. Right now it's not visible.
It's not visible. Just 1 minute. Let me share it.
Yeah, ma'am. Please take your own time.
This one. अब दिख रहा है?
हाँ दिख
हाँ
रहा
जी,
है
दिख रहा है, large scale plankton
plankton
culture.
cultures.
Yeah, it's visible, ma'am.
Okay. So just let me close the door one. So you can hear me and you can see the screen.
Yeah,
Yes.
ma'am.
All
Yeah.
right, wonderful. So now that you have heard, I think 3 talks by now on the importance of zooplankton, let's discuss some details about how to culture zooplankton on a large scale. I have been culturing zooplankton for more than 30 years, I think now, and it's really interesting, and eventually one gets a feel for culturing these species. Now, as I was answering some questions in the morning about the central role of zooplankton, you can see this figure over here, and this is where zooplankton is, right in the center of the food web. It connects all the material that comes up from this decomposing matter and from the growth of phytoplankton and takes it right up to other trophic levels such as fish and birds and other organisms. So it's a very important role. Zooplankton are also very widely distributed globally. So it's not a difficult thing to find them in local water bodies. They have high growth rates, short life cycles, and they can be used to verify many ecological hypotheses. Therefore, they are very useful also in answering and discussing details of evolutionary biology. Because in a short time, you can develop hypotheses and test them using zooplankton. But that's a talk for another day. They are also very sensitive to contaminants, and that's why the role in ecotoxicology. They are indispensable, absolutely indispensable as food for fish larvae, and that's their importance in aquaculture. And taxonomy, as I told you, it is changing all the time, especially with the input that we are receiving from molecular analysis and what we are learning that species that look the same need not necessarily be the same. So many different ideas are being generated in these fields. And zooplankton are absolutely, very, very important organisms for testing ecological hypotheses. Now, before we start culturing zooplankton, you might ask why. So suppose we take a look at the aquaculture aspect. This is the formula given by Shirota in 1970, and it is still in use today, where he discusses the gape size. Very easy to analyze because they consider the gape as a right angle triangle, and therefore you take the length and the width and the hypotenuse, and then you find out what is the gape of the fish larvae. And you see how the fish larvae gape changes depending upon the species, depending upon the age, and therefore you see that with an increase in the length of the fish, you see how the gape changes. It's increasing all the time. Now, once it reaches this level, a fish larvae will not try to choose a smaller prey because a smaller prey as fish larvae, they eat the prey one by one. So they individually pick their prey. So it is more energy. It is less energy to choose one cladoceran than to eat 500 rotifers, for instance. So if the gape will allow it, the fish larvae will normally prefer larger prey sizes. That is why you choose to culture rotifers only during the, I don't know, the participants can now see your application. It says, you can see me, right? I mean, the talk you can see. Hello?
Yes, ma'am.
You can see the talk and you can hear me, no
Yes,
problem.
yes, no.
Okay. So that is why fish larvae will choose to pick their food one by one and not filter it. And that is why it is better for them to feed on one cladocera rather than 500 rotifers, if the gape allows it. Now, in order to culture zooplankton, what is the basis? First of all, we need to have algal cultures. The algae should measure between 5 to 10 micrometers in size with a maximum of 20 micrometers. This is the size range which is normally used for algal cultures for zooplankton. There are several species that are used. For instance, in freshwater systems, you have species such as chlorella, Senedesmus, Chlamydomonas, Cryptomonas, Rhodomonas. In marine systems, you can use species such as Tetraselmis, Nanochloropsis, Chlorella, species of Chlorella which are marine, and Chetoceros. Culture medium. So we look at the culture medium. Now, which culture medium we should use for growing algae? Normally, all algae have got macro and micronutrients. They vary in minimal quantities. Unless your work is very specific, you can choose any culture medium of your choice, and it can be used for culturing algae easily in your laboratory. For instance, some scientists cultured 200 species of algae in just 2 different media, one for freshwater, one for marine. So it really does not matter, exceedingly, if you choose any one culture medium and use it effectively. During my PhD days in India, I was using Poole and Lorenzon's medium to culture chlorella. And in Mexico, in our laboratory, we have switched to Bowles basal medium, which is what we normally use. We also use the F2 Gillard medium, and we probably use some media such as the BG11 medium for cyanobacteria. Now, all these culture media, they provide nutrients in different proportions. They provide macronutrients and micronutrients. So the macronutrients, potassium, sodium, calcium, magnesium, phosphorus, nitrogen, and iron. And micronutrients are boron, zinc, manganese, copper, cobalt, molybdenum, and a few others. We normally add carbon to our cultures periodically. This carbon in several laboratories in Europe and United States are added by bubbling in the carbon, but we add just sodium bicarbonate, about 0.5 gram for 2 liters of medium. This is just the formula of the Bowles basal medium that we use. But in this formula, I would like to highlight one aspect that we normally follow in our labs. The point is that the media says that you should add 5 grams of sodium nitrate, for instance, to 200 milliliters of water. Then from that, you take 10 ml. Now, what we do, instead of adding 5 grams, we normally add 50 grams. And instead of taking 10 ml, we take 1 ml. So we add 50 grams of sodium nitrate to 200 ml of water and take only 1 ml. What is the advantage of this method? The medium that we prepare is more concentrated, and we can keep it for quite some time, a couple of months in the fridge without any problem, because there will be no infection of any kind in this medium. Now, because it is highly concentrated, the salt, and you use less at each time. So this technique allows us to prepare very concentrated media. We keep them in the refrigerator, and they are used over a period of maximum 2 to 3 months. We don't prepare for more than that. Are there any doubts until now?
No, ma'am.
Okay. Then you have very simple media that have also been developed, such as those by Achari and Menon in 1993 here in, I think, Sifri. It was developed, and it's a lovely idea because they use only potassium dihydrogen phosphate and potassium nitrate. There's nothing else that is there in the media. It's just a source, one source of phosphorus, one source of nitrogen, and they add a little bit of B vitamins. And that's all. And with that, they culture their algae. So you can see that it can be cultured in a very simple manner without any problem. And we have also cultured algae in one of our articles we have published by using commercial fertilizers. In this case, there is one which we buy over there. And these are just garden fertilizers. You use them in this proportion. And if you use them, the quality of the zooplankton which is produced is also not bad. So that is why we use these type of different media. Now there are three different phases in algal growth: the log phase, the exponential phase, and the asymptotic phase. You will see this in the next figure. So you have here the exponential phase starts here, and this is the log phase of the growth, and then it reaches an asymptote. Now, when do we harvest the algae? If you leave it for a very long time, hoping for much higher densities, you will have old algae that develop toxins and do not support zooplankton growth very well. So the best phase for the harvest is during the exponential phase and a little bit before that. For instance, here, if I were to harvest my algae, I would do it even here, so that I get good healthy algae rather than high densities of old algae. And once we harvest the algae, there are different ways to concentrate. Either you can centrifuge the cells, or what we do is simply put the bottles of culture in the refrigerator. And in that, within a period of 3 to 4 days, all the cells settle down. So once the cells are settled, you decant all the liquid, the culture medium, and you can use the algae for culture. You can also develop large-scale outdoor culture in 2 to 5 days, and after that, zooplankton is inoculated. How do you scale up a culture? If you see these figures, these are the batch cultures which are used for culturing zooplankton on a large scale. So normally we culture the algae over here in these type of recipients. You start with a very small tube and then you scale it up slowly. You put it first in 250 ml, and then when that becomes very green, you put it into 500 ml. And when that is very rich in algae, you then scale it up to 1 or 2 liters. When it comes to this level, we normally use a little bit of aeration. As you can see here, there is aeration. This aeration is only meant to put all the algal cells in motion so that they all can capture sufficient light and divide rapidly. Now, once you have the zooplankton like this, now the phytoplankton, sorry, you can now take this culture and inoculate it into prepared tanks outside. And within 2 to 5 days, these tanks are now ready to receive zooplankton. So this is what I explained to you. This is exactly what is written in this slide. There are other methods of culturing algae. One is a continuous culture and non-continuous or batch cultures. We follow this system of batch cultures in our lab. And continuous cultures are when you put the algae here and constantly you are taking the algae, filtering it out, and the water returns to the system. And then you have a continuous culture of algae. These are very complicated systems. These are normally used to test hypotheses rather than using on a large scale, because you have a lot of possibility of contamination with ciliates, with the other organisms in your culture. But that doesn't matter much to us in a batch culture because it's just to feed zooplankton. Obviously, if you have too much of contamination with protozoans, that won't be good for offering as food to rotifers because there will be a lot of interspecific competition. Then you also have the feedback culture where you have the algae and you have zooplankton. And then the zooplankton eat the algae, the water is filtered, and after some treatment, it goes back into the algal culture. These systems can be used. They can last for several years, but they need a lot of maintenance. So for our needs, we normally follow the batch culture system. What are the other problems that you could see when you are trying an algal culture? First of all, you need to have an appropriate culture medium. You must be sure that the culture medium, the algae are adapted to this medium and they can grow well. You must be able to provide sufficient light and aeration. Bacterial contamination can be controlled using penicillin or streptomycin. And the growth of different species of algae at the same time is a big problem. So that you really must avoid, because eventually what will happen is one species will outcompete the other and you will not have any, for instance, let me take the example of chlorella and Senedesmus. Chlorella measures about 5 micrometers, very appropriate for rotifer cultures. Senedesmus measures around 10 to 15, 20 micrometers, very appropriate for cladoceran cultures. Now, if you are mixing both of them, rotifers won't grow well on Senedesmus. So you need to provide them chlorella. If that is the case, then if your culture is contaminated, eventually chlorella will be outcompeted by Senedesmus. So you are thinking that you are providing actually chlorella to your zooplankton, but you are providing Senedesmus and your rotifers are not growing. So you need to be very sure that your algae are growing well at all times. The presence of unwanted zooplankton also occurs in large-scale cultures, for instance, such as deloids, but it is not very difficult to separate zooplankton from algae, because if you use a mesh of about 40 micrometers, you can easily separate any zooplankton from any algae that you are growing for zooplankton growth. Now here we have the image of a cyanobacteria anabina. And if you have cyanobacteria in the cultures, also you won't be able to allow for good algal, good zooplankton growth. So you need to be careful and need to be vigilant, always looking at the algal cultures and not believing that they are all well and everything is nice because it looks green. Another medium which is used, which was developed by Kilham et al., is the combo medium. And the combo medium is a freshwater culture medium for algae and zooplankton. So you can use this medium also for growing algae as well as zooplankton. How to filter the zooplankton? In the case of algae, I would always recommend, if you don't have algal cultures in your lab, please request somebody to give you a small sample of algal cultures. It is not impossible to separate algae from a water body and start your own culture, but it will take you up to 6 months to establish. It's much easier to start off with a small request of an algal sample, and everybody is normally willing to oblige. In the case of zooplankton, it's not so difficult. In the case of zooplankton, you go to a nearby pond or lake or reservoir or river or wherever you want to work, you filter. So what we do is we filter about 80 to 100 liters of water through these meshes. We prepare the meshes using these tubes, and you cut the tubes to the size you want and you stick the mesh in between the tubes and you can prepare the meshes. These long conical tubes, normally we get them prepared by experts or you buy them in the market. These are also very nice and fancy, but it's not really obligatory. I mean, it's very convenient, yes, if you have to filter several thousand liters of water, but since you are just filtering only 50 to 100 liters, if you have a small piece of mesh, it's more than enough. What mesh size? This is an important point. What mesh size should we use? We normally use 20 micron meshes to test for phytoplankton and about 50 to 60 microns for zooplankton. So, and some people do not even filter zooplankton. They simply bring it directly and fix it and do their studies. But we normally filter the zooplankton through a 50 micrometer mesh. Now, we have heard this morning about the importance of zooplankton cultures. They are very useful for fish larvae. They have very high nutritional value. The disadvantage here in zooplankton culture is that they need to be used fresh. You can freeze zooplankton. You can use other forms of preservation of zooplankton also, but this greatly reduces their nutritional value. So the biggest point to remember when you are trying to culture zooplankton for fish production is that the zooplankton production must be planned very well. So you must be able to say that, all right, if I am going to get my lot of fish after 15 days, then I must start my culture today. So that kind of thing you must be able to establish, and that can only be done by preliminary studies in your respective laboratory. Why do zooplankton have such high growth rates? They have a short generation time. They have small body size, half a millimeter to about 4 millimeters max. Especially in Cladocera. In copepods, they can be much larger, about 10 millimeters also. Then we have lifespan 15 to 40 days in rotifers and cladocerans. In copepods, they can be sometimes much longer. Some species live less, for instance, 3 weeks. Some species live more, until 3 to 12 months. They have very high net reproductive rates and an early age at first reproduction from 18 hours to 96 hours. So these factors all add up to the fact that they can have extremely high growth rates. They can reach very high densities. And in this way, we can all utilize zooplankton for many different purposes. As I showed you in the previous slides in the last talk, rotifers and cladocerans normally reproduce parthenogenetically and sexual reproduction. Now, sexual reproduction, many people say that it occurs during unfavorable conditions. Hypotheses are varying, hypotheses are being tested, and some people say that sexual reproduction occurs all the time in order to maintain an egg bank. Regardless of the situation, these resting eggs or ephipia, resting eggs in rotifers, ephipia in cladocera, and dormant embryos in copepods are extremely useful. So you must try to preserve them. Do not throw away the bottom part of your culture when you are changing the culture, because it is very important to keep it and freeze it. If you freeze it, and if you have to go away for a holiday, you don't have to worry. When you come back, you take out that filter paper and put it up for hatching. Eventually, some individuals will hatch. An article more than 50 years ago by Boras, I think Boras and Boras, they said that Brachyonus caliciflorus is the fastest growing metazoa. It has extremely high growth rates. And that is why, as Dr. Sarma also must have told you, very useful in ecotoxicology. In the case of Moina, also has extremely high growth rates. And we are very fortunate to live in tropical countries where we have plenty of Moina around us. Now let's take a look at copepods. In which kind of system would you try to culture copepods? Copepods are commonly found in freshwaters also. However, in freshwaters, you have the choice of using rotifers and cladocerans, which have higher growth rates and which are a better food for fish larvae. But in marine systems, cladocerans are very poorly represented. Rotifers are there, plenty of rotifers, which as we will see. But so in marine systems, we would like to use mostly copepods. Now, which copepods do we use? We use a species such as calanoids and herpacticoids are normally used, for instance, Tigriopus or Acartia or Paracalanus. These copepods are very useful because they have very high levels of fatty acids. Despite their diet, they could be feeding on detritus and yeast, but they are capable of synthesizing these fatty acids, and therefore their nutritional quality for fish larvae is exceedingly good. Now here is another image of the life cycle as I showed you in the morning. You have from eggs, naupliar stages, copepodite stages, and then you come to the adult stage. And here you have the productivity in semi-extensive, semi-intensive, or intensive cultures and how many can be produced. And this is in a course manual which has been produced here in India by the Central Marine Fisheries Research Institute. Now, most copepods never have a parthenogenetic phase. They need males and females for starting the culture. But some species are parthenogenetic, like I told you, Elafoidella. Grandidieri is a herpacticoid, parthenogenetic, and extremely easy to culture. You don't even have to look at the culture for months. If you reach a well-established culture in an aquarium, you just Keep feeding it, and once in a month or once in 3 months, change the culture, it's good enough. They grow very easily. But calanoids and cyclopoids, and you need to take care of them for maintaining good cultures. You can feed copepods algae. You can feed them alfalfa infusion. Some people in Mexico have suggested that even what do you call it? A juice of spinach can also be used. You can feed them rotifers. You can feed them commercial diets. Try what works best for you in your lab. Now, the presence of male indicates that the culture is in a phase of sexual reproduction, and the growth rate will definitely decrease. So the growth rate of your culture is going to go down if you find males in the system. You have to be careful. Cladoceran males have got a long lifespan, but rotifer males cannot live for more than about 24 to 36 hours. They measure very small, 10 micrometers. They look like ciliates. They do not grow much. They do not eat at all, like Dr. Sarma told you. And in the case of Cladocera, the males are about 1/3 or half the size of the females, and they feed and they grow also. But if you find males in the system, then you will not be able to find a high growth rate, because there will be an investment in Ephipia production. I think Dr. Sarma has spoken to you about the importance of the egg ratio. This was one of the best contributions of Edmondson, very important contribution of Edmondson for aquaculture purposes. And it clearly indicates that if the proportion of eggs to the females is low, the conditions are not very good. So you can easily measure the egg ratio in your large-scale cultures. As it declines, you will know that it is time to harvest everything and start the culture again. You can see over here, in the initial phases, all the females have a lot of eggs, so there are many eggs present. With time, it goes down, like you see over here. And you can see here that in the controls, it decreases like this, but in the presence of competitors such as Daphnia pulex or Moina macrocopa, the decline is more severe and rapid. We also have cultured rotifers in Nahayote. What is Nahayote? I think that many of you are aware that in Mexico, most people eat tortillas made out of maize or corn. Now, the corn has to be processed using calcium. It is not to be eaten directly, because if it is eaten directly, the nutrients, the proteins in the corn cannot be absorbed by the human body. So Mexicans have developed a very traditional and very old method, more than 1,000 years. They use the corn by digesting it first with calcium. Then this water is thrown away. This water has got a lot of nutrients, and these nutrients can be used for the culture of rotifers. For instance, you see over here, you have the Nahayote water. You can see the growth of Brachyunus caliciflorus only with algae. And you see the system here only with algae. And here you are providing it Nahayote water with algae. And here the square blocks without any algae, only Nahayote water. And you see that it reaches a peak much earlier. Higher growth rates here with 4% of Nahayote water. Now, obviously, you don't have this water here in India, but you can use any other type of organic matter also for the growth of rotifers. Here with yeast alone, they don't grow very well, as you can see, but they can grow with a mixture of algae and yeast. And the advantage is that it is very costly to produce algae. It takes a lot of effort, but yeast is much easier. So if you mix algae and yeast, you can have a high density of rotifers with lower cost. You see the population growth of Moina now. Moina at 20 degrees and 30 degrees on different diets. And with chlorella and with microcystis, obviously microcystis is a cyanobacteria, doesn't grow very well, but with chlorella, you reach high densities in 10 to 14 days. In the case of Daphnia carinata, the growth rate is much lower because it has a very long time before it reaches the exponential phase of growth. And by 28 days, so the R value will be much lower in the case of Daphnia as compared to Moina. If you compare the temperature also, for instance, you see over here, Moina reaches a peak of about 80 individuals, and at 30 degrees, it reaches much earlier, within 5 days, about 250 individuals. So you must pay attention to these details and do small experiments, preliminary experiments, in order to determine all these details about the species of your interest. In the case of Daphnia carinata, it reaches 20 degrees. At 20 degrees, it reaches about 200. But at 30 degrees, it doesn't grow so well and reaches a peak of no more than 100, 120 individuals. Now, zooplankton are very, very important, as we have all seen. Mostly a large proportion which is useful in aquaculture are rotifers, cladocerans, and copepods. So if you are able to culture all the three, that would be just ideal. In this case, this species, Brachyonus rubens, is a very good food for fish larvae. However, you have to be very careful, because if you have Brachyonus rubens in a culture with Cladocera, the cladoceran density will go down, because Brachyonus rubens tends to be epizooic. It sits on top of the cladoceran and makes it slow, and eventually the species doesn't have enough energy and it will die. So you have to be careful which species you are choosing. If you choose Brachyonus rubens as food for fish larvae, excellent, because it grows very fast, it grows on a mixed diet, and all fish larvae can feed on it very well. So the size is appropriate. You can see it has no posterior spines, very small anterior spines. So it doesn't provide any kind of resistance to feeding by the larval fish. Similar situation here with this species, which is known as Brachyonus dimidiatus. Brachyonus dimidiatus is also found in India, especially in spirulina cultures. So there it is a big problem. However, it is a nice species to try and culture, especially for saline systems. It can tolerate salinities from 10 to 20 grams per liter quite easily, quite well. Now, rotifers and cladocerans are widely used. Rotifers, which are most useful for aquaculture, measured between 150 to 300 micrometers. Most of them are brachyonids. In freshwaters, we use Brachyonus calciflorus and Brachyonus rubens. In marine systems, we use Plicatilis, Brachyonus plicatilis, Brachyonus dimidiatus, Brachyonus rotundiformis, etc. Brachyonus plicatilis can reach densities of more than 2,000 individuals per milliliter. So just imagine, you can do the math. Just 1 milliliter has 2,000 individuals if you are able to culture it well. Even if you are able to culture it with very less skill and expertise, you can easily get up to 200 to 500 individuals per milliliter. And freshwater fish larvae need 2,500 rotifers per larva per day. So if a marine fish larvae needs about 100,000 rotifers in the first 2 weeks of its life, we think that it is a whole lot of rotifers. No, no, no. You see that in just 1 ml, you gave them 2,000. So if you do the math and see, you'd hardly need about 10 ml, and you can get all the rotifers that you need for your cultures. You can use different kinds of media. I just spoke about it. Yeast, bacteria, algae, nahayote, organic waste of any kind can be used. And you must try all this in your system before you start establishing it. Preliminary experiments are highly recommended. And pay much attention to the egg ratio. It's a very good culture of the health of the culture. It's a very good indicator of the culture's health. Now, after taking, if you have the cultures in the lab, now you need to inoculate the ponds. Outdoor ponds with a capacity of 200 to 2000 liters are used. These are filled with tap water, left to dechlorinate for about 1 or 2 days. You add commercial fertilizers, or you can add farm and poultry waste. Farm and poultry waste kind of, induces a quick, rapid growth of algae, but it can lead to problems due to contamination by parasites or by other diseases. So you have to be careful what is the final end product is going to be used for what reason. Depending upon that, you will choose whether you want to use a commercial fertilizer, or you want to use some special media, or you just simply want to put farm and poultry waste. I have cultured zooplankton using farm and poultry waste and oil seed cakes, mustard oil seed cakes. Excellent growth of zooplankton and extremely high densities. I was also talking about this in the morning, about the nutritional quality, and you can see that Moina and Daphnia have a much higher and much better quality in terms of proteins, in terms of ash content as compared to Artemia. Artemia is more expensive, more difficult to maintain, and if you can establish cultures of zooplankton, it would be excellent. Now here there are several studies in the literature which clearly indicate that copepods have got very high levels of these fatty acids. And when they have these high levels of fatty acids, despite their diet, it's not important what species what diet they are being given, they are able to generate these fatty acids. So this is extremely good for fish larvae. In the case of rotifers, you can see that when you provide them with yeast, their nutritional value decreases. For instance, they have less proteins as compared to rotifers cultured on chlorella. They also have less lipids, and they have less ash, but that is not important in the case of rotifers to that extent, but they have less proteins and less lipids. So it is important to give rotifers algal diet at least just before 1 hour before feeding them to fish larvae. Now, how to start a culture? For those young people who have never cultured before, these are just some tips, but many of you probably already know these details. First of all, we need a good microscope. You need pasture pipettes. You need meshes from 50 to 200 micrometers. It's good to keep a wide range of meshes because you can use them to separate species if they are mixed with each other, if they get contaminated. Then we use 5 to 50 ml glass and plastic containers. After that, you can use 1 liter, 2 liter glass bottles of any kind. That's not important. Actually, it's not necessary that you invest in a whole lot of very fancy 1 liter and 2 liter beakers. You can use jam bottles and pickle bottles. They are very, very useful, perfect for culturing zooplankton. No problem at all. And people who publish in Science and Nature also use these jam bottles for culture. So don't think that the jam bottle will make a difference. It is your idea and your effort in the zooplankton culture as such. Then you can use light. We use 12 hours light to 12 hours dark. We use different temperatures depending upon the species of your interest. It's a good idea to test when you go to the field. Take measurements of pH, temperature, conductivity, and simple factors, and try to replicate that in the lab so that your zooplankton are already adapted to it and they will grow better. You can collect the samples from local water bodies, identify the species. If you cannot identify the species, there is no problem. Take plenty of pictures with your cell phone, send it to experts, and most experts will be willing to oblige with the name of the species. You isolate 50 to 100 individuals of each species. You start a clone with one individual, take 5 different recipients, start clones, and after that you keep a stock. It is better to use a female with eggs, rotifer with eggs to start the clone, because then you will be more motivated because these eggs will hatch, the next day the culture will grow and you will be very happy. Taking a look at the water quality, just give me a moment to get a little water to drink.
हाँ खत्म हो गया.
Thank you. I'm here. I'm back. Okay. So water quality determines the culture health. You need to have very good quality water. There are 2 or 3 different options that I normally suggest. Number 1 is use simple tap water, dechlorinate it, and add these EPA salts. These salts make the water moderately hard. Thank you. Thank you. These salts make the water moderately hard, and these are simple salts, very cheap: sodium bicarbonate, calcium sulfate, magnesium sulfate, and potassium chloride. You can add these salts to a dechlorinated tap water, or you can add it to distilled water, or you can use directly filtered lake water. If you filter the lake water, if the lake is good quality water, then try to use that. And when you start the culture, you can start adding different proportions. For instance, you isolate the organisms directly into the filtered lake water. After 3 or 4 days, make it 75% lake water, 25% medium, and so on. Like after another 4 days, 50%, 50%, and so on. So in this way, the organisms are slowly adapted to these conditions of culture. We have maintained here, I say 40 species, but I think that by now we have many more species. I think more than 60 to 70 species we have cultured using EPA medium and chlorella or Senegal. So you don't need many different type of media, but you need to adapt the organisms slowly. In the case of copepods, if you are starting a copepod culture, these organisms are often very cannibalistic. They eat their own nuclei if they don't like any other food. So under those circumstances, you should try to separate the adults from the nuclei using a mesh. You should try to separate them. So we separate them using a 50 micrometer mesh, and these can be used to the nuclei will come down and they will be protected from the adults, and you will have a faster growth rate of your copepod culture. What do we feed? Cladocerans are best fed a mixture of algae, bacteria, and yeast. You can also filter water directly from a lake. Filter it through 20 or 10 micrometers so that you don't get other lake organisms, and offer this as food to your cladocerans or copepods or even rotipods. Now, many cladocerans tend to get trapped in the surface layer while changing the cultures. In order to prevent this change, you can submerge the mesh in water and never expose it to air when you are changing the culture. Then your organisms will not get trapped on the surface layer of the water column. Now, when the density of your culture increases, you can increase the volume. Normally, if I start a culture with one individual of a rotifer, I would put it in about 10 ml or 20 ml. And when the density increases to 50 or 100 individuals, I try to increase the volume. So slowly one increases the volume. If you put one individual of rotifer or one cladoceran in 1 liter, it's eventually going to die. It won't survive well. So please try to use one by one. On the other hand, you have to also keep a very close watch on the color. If the color of your culture is clear and yellowish green, then you will have a very good culture. But if it is opaque, then it is not a very good culture. So please pay attention to these details. And I'm sure that you will be able to establish your own cultures. You really don't need to go to any laboratory. You can go back to the pond, get some more animals, and try to start again. In large-scale cultures, as you can see in this image over here, our students are inoculating these type of tanks. Large-scale tanks are being inoculated here. So what we do is first we fill the tank, we leave it for 2 to 3 days to dechlorinate, then we add the phytoplankton or we inoculate it. When we test the transparency using a Secchi disc, you inoculate the rotifers. In these large tanks, you need to have 1 to 2 liters of a culture with at least 20 individuals per ml. In case of rotifers, in case of cladocerans, you need 1 to 2 liters of a culture with at least 5 individuals per ml. And in the case here, we also need zooplankton. If you want to know, this is a very nice formula which was suggested by Jingren in 1991. If you want to know if the zooplankton is enough for your fish larvae, you filter 50 liters approximately of water and put it into a glass tube like a measuring cylinder, 100 ml measuring cylinder. And then in that, if it reaches a 2.5-inch diameter, of a 2.5-inch diameter tube, and if it reaches about 6 to 8 millimeters of zooplankton, that means that your culture of zooplankton is enough for 500,000 to 750,000 larvae per hectare. So if you follow this nice formula by Jingren, you will know whether you have enough zooplankton for the fish larvae that are there in your pond. When do we collect the fish larvae, the zooplankton? You collect the zooplankton when the transparency is like it goes down. You can see almost it's crystal clear. That means there isn't enough food. If there are blooms of rotifers or cladocerans, which you can see by the reddish color, collect it because you will not be able to keep it. They will eventually die. If the weather conditions are bad, for instance, if these species are adapted to 30 degrees and suddenly it starts raining and the temperature comes down to 25, they will also die. So you collect them at that time. If the egg ratio declines and is low, it is less than 0.2, it's a good time to filter and collect all the rotifers and offer them as food to the fish larvae. And also if you have a lot of contamination with other organisms, such as ciliates, they won't grow very well. So you should collect your organisms and offer them as food and start the culture all over again. Do not discard the resting eggs and ephipia. As I said, they will be very useful. They will eventually hatch and you will have your cultures again if you can keep these ephipia and try to hatch them. They can be used to restart the culture. The hatching rate is not high, but never mind, you can still use, and with 1 or 2 individuals, you can get your culture back. What are the problems that we will face, or we could face, or we have faced, or we always face in zooplankton cultures? Each species must be kept very carefully with its own mesh. Do not use the same mesh for many species. That will avoid a lot of contamination problems. Then contamination caused by ciliates or other species of cladocerans and rotifers or as plankton or flatworms are quite difficult to deal with, but you can deal with ciliates, for instance, by filtering through different sized meshes. If the ciliates are rather small, then the mesh size alone will allow you to clean the culture well. If you have other species of cladocerans and rotifers mixed, you can use meshes. For instance, if you use 150 or 130 micron mesh, you can separate rotifers from cladocerans easily. If you have predatory rotifers, such as Asplankna, this image that you see over here, if you have this Asplankna in your rotifer culture, be very careful because your culture will not survive more than 5 days. Asplankna will finish off each and every brachionid. So you have to keep looking at your culture. And Asplankna, fortunately for us, is quite large. So it measures around 800, 700 micrometers, which is thrice the size of a brachionid, depending upon which species you're culturing. So sometimes meshes can be useful, but you need to keep a close watch. A good microscope is essential for your laboratory. And then you have also species such as flatworms, Mesostoma, Stenostomum. These also feed on our zooplankton, on our rotifers, cladocerans, etc. So if you have these species in your culture also, I think the best way for separating Mesostoma or Stenostomum, if these are less, you can pick them out. Otherwise, it's better to pick out 100 or 200 individuals very carefully of your species of interest, pass them through different washes and repeatedly, for instance, 2 or 3 times. Eventually, you will have a clean culture and you can start the culture again. Salt shock can also help a lot. For instance, if you give a 10 grams per liter of sodium chloride, you can reduce the ciliate load, but this you should do only for about 1 minute or so, because otherwise the cladocerans or rotifers are also very badly affected. These are all freshwater organisms that we are talking about. And vice versa, if you're working in marine systems, you can try a freshwater shock. For instance, another technique is to learn about the ecology. For instance, I told you that Brachynos rubens is epizoic on cladocerans. So if you have a Brachynos caliciflorus culture and you want to separate the rubens from it, put a few Daphnia, put a few Moina there. Eventually, all the rubens will stay on the cladocera, and slowly you will be able to get rid of the contaminant species, which is Brachynos rubens. Thank you very, very much for your kind attention, and I'm willing to answer any doubts or questions that you may have.
Ma'am, I just ask about we are all here in India, we are also doing the prawn culture.
Mhm.
Is there any difference for the fish culture and the prawn culture?
No, for prawn culture, actually, the expert is Dr. Sarma. You should talk to him. He has a lot of experience on prawn culture. But for prawn culture also, you need to culture the zooplankton in this manner, in very big tanks, and then offer them to the species. Also test the densities in ponds. But tomorrow, please kindly ask him. He has experience and he can answer your doubts with regard to shrimp and prawn culture. He worked on a shrimp farm for quite a number of years.
Okay, thank you.
Mhm. Any other questions regarding? Yes, please.
Ma'am, unmute yourself.
I can, yeah.
Maheshwari, ma'am.
Yeah.
Madam, I want to know, is there any relation between the zooplankton culture and climate and uphealing in the lab, or as well as in the natural condition?
Zooplankton culture, climate, and I couldn't get the last.
Upwelling in the in lab or as well as in the natural conditions.
No, the point is that if you are culturing in the lab, you can maintain the temperature condition.
Yeah.
But if you are culturing outside, you need to know during which season. For instance, if you are in South India, the temperature is more or less the same throughout the year, right? But in North India, it becomes extremely hot. For instance, in Delhi, you can reach temperatures of 45 degrees in the summer and 15 degrees in the winter. That type of temperature drop is very difficult for zooplankton to grow in natural conditions. So you need to know what is the temperature, and it would be best to maintain in a kind of a greenhouse or somewhere where the temperature can be kept steady.
Thank you, madam. There is one another question from my side. That is, you said in your talk that in lab condition you use 12 hours light and 12 hours dark conditions for the culture. Is it so important, or we time point is the different, or the hours are the different, 16 or 12 or 10 or
it's not that important. It's just a question of also trying to save on electricity, right? And the zooplankton don't need that much electricity or that much light as such. But if you have phytoplankton cultures, obviously you need to have 24 hours light.
Okay.
In phytoplankton, we normally keep 24 hours. People are there who keep 16 and 8 also. But
Yeah.
we try to keep 24 hours light when we grow the phytoplankton. For zooplankton, it doesn't really matter so much.
Okay. Thank you very much. Thank you.
Welcome. Any other questions? Dr. Sarma is here right now. If you would like to ask your question on shrimp culture, he will be happy to answer, I think. Dr. Das has a question because they are doing some shrimp culture.
Is there any difference for the fish culture or prawn culture? We use the zooplanktons, which is better for the prawn culture.
No, it's like this. For shrimp, which for freshwater,
So your voice is not.
is it shrimp?
Not very clear, sir.
Are you talking about the freshwater or marine?
Freshwater, sir.
Freshwater. Yes, the shrimps require zooplankton, rotifers, especially only first 3 weeks. After that, you have to use balanced diet. Otherwise, the growth will not. And also, the diet, what called pellet diet, it must be age specific.
Okay.
So there are specific protein composition, lipid composition based on the age of the larva, age of the shrimp. In case of marine shrimps, after 20 days, we use a trash fish. So you collect the fish from the non-edible fish, collect them and boil them in the water, and then take them into small pots and filter them and pass through 1 millimeter or 2 millimeter mesh size, and whatever falls below that you collect and give them. That is a very good one. But there's one problem: if you don't change the water frequently, fungal infection will take place.
Okay. Okay. Thank you. Thank you very much.
Any other questions? Any other questions from the participants? You may please ask. I think so. There is no more questions. So the lecture was indeed very interesting.
Hello.
Hello. Are you able to hear me?
Yes.
Are you able to?
Unmute all participants.
Yes.
Okay, fine. Now I started from again. The lecture was indeed very understanding, very impressive. And to propose a vote of thanks to Dr. Nandini Sarma, I would like to call upon Mr. Satish Ramteke, Assistant Professor, Department of Zoology, NPG College of Science, Raipur. Over to Satish Ramteke.
Thank you, ma'am. Good afternoon, all respected dignitaries, chief guests, seniors, and all the participants present in this online international webinar on plankton biology. I am grateful to Dr. Nandini Sharma, ma'am, who presented her lecture on the topic large-scale culture of phyto and zooplanktons. Her lecture contained minute details about the life cycles of various zooplanktons and its critical role in their culture. It also she also told about the ecotoxicological studies and their relation to fish farming. She also told us about the role of tortilla in Mexican food culture, and their role in culture of rotifers. She told us about the various techniques for cultivation with minute details therein. On behalf of the Department of Zoology and the entire family of Government Nagarjun PG College of Science, I heartily thank Dr. Nandini Sharma, ma'am, for her informative and enlightening lecture. Thank you, ma'am.
Thank you very much.
So, on behalf of all the members of NPG College of Science and all the participants, I extend a really hearty vote of thanks to our eminent speakers, Dr. S.S. Verma and Nandini Sarma. Today, we had a golden opportunity to hear a wonderful lecture from both of you, and it was very interesting, and your interesting research findings, and this will surely going to be a very useful in the research of participants of phytoplankton as well as zooplankton. So with this, we come to end of today's session. Tomorrow we shall be meeting again for the next day of our webinar at 10 a.m. Thank you so much, sir and madam. Thank you to all the
Thanks, ma'am.
participants.
Thank you very much, ma'am.
Thank you, sir. Thank you, ma'am.
Thank
And
you.
thanks to all the participants too.
Okay. Thank you very